Hello everyone, welcome to your 1000 MCQ sessions for NABARD Grade A Phase 1. Yes, we are completing almost 40 questions per session and we are again back with the session. This is your part 8. Okay, so let's start our session with the first question. A crop grown primarily to improve and protect soil from erosion through their ground covering foliage is called as what? We have already seen this question, guys. Come on, tell. Guys, these questions we have done. Wait a minute. Let me just bring up good questions. Yeah, the seedlessness in fruits is called as what? This is our first question. Sorry guys, this is our first question. The seedlessness in fruits is called as what? Try to answer with me. More answers you will try to give. I will also move with the questions very fast. More questions you will try to answer. I'll move quickly. The seedlessness in fruits is called as what? Parthenogenesis, parthenocarpy, apomixis, explant, or none of these. So the answer here is A, which is parthenogenesis. So when we talk about parthenogenesis, no, it's not parthenocarpy, it's parthenogenesis. Okay, so in some plants fruit develop parthenocarpally, still they produce viable seeds. So parthenocarpic fruits are nothing but where your seedless fruits is developed, but the process is not parthenocarpy, it is parthenogenesis. So don't get confused. Okay, where seeds are not there, it's not parthenocarpy, it's parthenogenesis. Okay, next, blue color certificate is issued to which of the following class of seeds? Breeder seed, certified seed, registered seed, foundation seed, and nucleus seed. Blue color certificate is issued for which of the following class of seed? Breeder, certified, registered, foundation, nucleus. Certified, are we clear? Are we clear, guys? Is it? Yes, it's correct. Certified seed. It is a progeny of foundation seed. What do you mean by progeny? In the sense, first is nucleus seed, then is your breeder seed, then is your foundation seed, then is your certified seed, then is your registered seed. Okay, so certified seeds progeny of foundation seed. The color is blue, or don't get confused, it will also be azure blue. तो डर नहीं जाना है, it's the same. Azure blue as blue. Next, select the correct statements on the classes of seed. Nucleus seed are produced directly, or nucleus seeds are product directly by the originating plant breeder at research institution. That means nucleus seeds are produced in the research institution directly. Breeder seed is also known as mother seed, and foundation seed are genetically 98.9% pure. So the answer here is D, which is 1 and 3. Breeder seed is not known as the mother seed. Breeder seed is not the mother seed, it's the nuclear seed which is the mother seed. So that is why your correct statement were asked 1 and 3 is correct. Your third statement is wrong because breeder is not a mother seed, nucleus seed is mother seed. Yes, you are right, 1 and 3. Foundation seeds are 98.9% pure and nucleus seeds are yes produced in research institute only. The main purpose of nucleus seed is to create progenies, that is to create the breeder seeds. Okay, so when we talk about your breeder seed, it is overall progeny of nucleus seed, and generally it is under the supervision of plant breeder institutions, state agricultural universities, etc. So when I talk about your mother seed, it is your nuclear seed. Next, certification is not required for which of the following seeds? Nucleus, breeder seed, foundation seed, certified seed, none of the above. Certification is not required for which of the following seeds? So certification is not required for A, correct answer, very good wisdom. It is not required for nucleus seed. So when you are considering nucleus seed, these are handful original seeds which are known for their particular variety of maintenance and purification by original breeder. So therefore here these are having highest genetic purity and physical purity, which makes them of best purity and which is genetic and physical. Now one of the most important thing is that their purity and quality is ensured by several inspections, and that's why if you see, the certification is not required for these seeds. Okay, so A is the correct answer. Before moving to the next question, okay, I will tell you. Before moving to a next question, yes, genetic purity of nucleus seed is your 100%. Now before moving to your NABARD next expected 1000 question, so one thing I'll tell you, if you're preparing for NABARD Grade A Phase 1 examination, there would be a question before you that in this last 10 days, how you are going to revise for the merit section especially. We all know two sections are there, qualifying section and merit section. In merit section, definitely you will be thinking about GA, ARD and ESI. What should I plan and how should I plan? Overall, what to do related to revision for ARD static, what to do revision for current affairs, which is ESI and GA, 60 marks, GA for 20, ESI for 40, ESI completely PIB government scheme will come. So what to do for that? So I hope you know that 1000 plus MCQs which we are trying to do on YouTube. I already told you, we have launched a dedicated course for NABARD also now, which is NABARD Grade A 2024 Phase 1 Merit MCQ batch. So if you go on our Oliveboard website and if you go on dashboard here, already 47% off is going on all our courses. So just go on the dashboard, check out here at the first row all RBI courses are there, then SEBI courses, and then fourth row is your NABARD row, in which your first course only is the well-known course NABARD Grade A Phase 1 Merit Section MCQ batch. In this batch, we are again giving 2000 MCQs in this last 8 days. So minimum 20 to 30 lectures are only there, but each lecture, it is like a marathon. So each lecture is like a marathon. In that marathon lecture, almost we will cover more than 50, more than 100 MCQs, and we'll make it sure that 2000 MCQs are covered here. What is provided in this? So 20 to 30 lectures, minimum lectures will be there. We are more concentrating in covering the quantity of MCQs, not on quantity of lectures. Here it will cover following things: 500 MCQs for ARD static and ARD current affairs, 1000 MCQs for PIB government schemes and current affairs associated with Ministry of Agriculture, 500 MCQs for complete general awareness, which is for 20 marks. So overall, again 2000 plus MCQs, and all these MCQs will be covered before your 1st of September. So 1st of September, before that we are covering this. Some first initial lectures will be recorded and rest of the lectures will be live. If you are a student, you are having panic, or if you are really scared to death for this NABARD Grade A Phase 1 exam, especially for merit section, consider that qualifying section में एक 2 mark का cutoff होता है. The cutoff is very less, so you can clear that definitely. What about the merit section? The overall cutoff is out of 100, GA 20, ESI and ARD 40 each. 100 marks में से cutoff कितना होता है? Past previous year the cutoff has been between 40 to 55 marks. So how to get this 40 to 55 marks? Definitely you should go for this course, and this course is, what is the cost of this course? It is just at 1200 to 1300 rupees. Just check it out. Okay, so 47% off is going on. 24499 is the price of MCQ batch. You are getting it just for close to 1000 rupees only. So best course to go for, because here minimum lectures are there. In this minimum lectures we are providing you 2000 MCQs for all merit subjects: GA, ESI, which includes PIB government scheme, and ARD static and ARD current affairs, everything. So in this way, definitely go and check out the course. You can see the course at the dashboard only. Just go on dashboard. Once you go on dashboard and scroll a little bit, at bottom all NABARD courses are there, and this is your NABARD first course only. Here you will see, that is NABARD Grade A Phase 1 Merit Section MCQ batch. The batch is starting day after tomorrow, that is 22nd, and it will finish on 28th or 29th of August. Okay, so in this 7 to 8 days only we are covering 2000 MCQs. It is nothing but all marathon sessions are there. So don't miss this particular course as well as lectures. Now one more thing, in YouTube we don't know that till how long these lectures are going to on, or will this continue or not. But the same quality of questions we are going to bring you for your these sessions. So make sure you are definitely there in this course. Check the detail of the course, and if you are having any doubt, connect with our counselors. Okay. Next. National Seed Research and Training Center is located at which of the following? Is located at which of the following? Varanasi, Jharkhand, Delhi, Sikkim and Pune. National Seed Research and Training Center is located at which of the following? Varanasi, Jharkhand, Delhi, Sikkim. The answer here is A, that is nothing but Varanasi. So, which is this training center? National Seed Research Training Center, Varanasi. The institute has been notified as a Central Seed Testing and Referral Laboratory, that is CS. It's not audible. Battery thoda kam tha. No probs. Okay. So what I was saying was, yes, it's a good initiative indeed, this 1000 MCQ session. But the problem is that it's a free session, so we cannot continue for very long. We cannot even guarantee you all the questions, which we will definitely try. But one important thing is that in the course which I just showed you, that is NABARD Grade A 2024 Phase 1 Merit MCQ batch, in that it's a course and it's at lucrative price, 1000 to 1200 based on the off. Now 47% off. For regulatory bodies, the offer is never above 35% or 40%. Now you have an opportunity based on Raksha Bandhan. So definitely go and check it out. It's a great course. In this last 10 days, those people who have anxiety related to NABARD Grade A Phase 1 exam, my guarantee that आपके पास confidence आ जाएगा. क्योंकि 2000 MCQs, when you will practice, the teacher is teaching 2000 MCQs, definitely you will get a hang of it. Marigoda, that's the answer. MH Marigoda. And Marigodan Halli, Humbegowda Marigoda is known as father of horticulture. Okay, so this is the correct answer. Which of the following methods are used for asexual propagation? Cutting, layering, budding, grafting, all of the above. So the answer is E, all of the above. What are the asexual reproduction or vegetative propagation, cutting, layering, division, budding, grafting. Okay. Scientific name of cotton is Ozyra sativa, Triticum avestium, Z. maize, Sorghum vulgare, and Gossypium. Ozyra sativa, Triticum avestium, Z. maize, Sorghum vulgare, and Gossypium. So the answer is E, which is Gossypium herbaceum is the scientific name of cotton. Okay. Remember this scientific name, very important. Up till now, cotton scientific name is not asked. Indian Constitution mentions article under directive principles and state policy. This article talks about to organize agriculture and animal husbandry by state, that is responsibility of the state to organize agriculture and animal husbandry on modern and scientific lines. Which is this article? Article 43, 46, 47, 48 and 49. So the answer here is your D, which is nothing but Article 48. Article 48 for Constitution, which is one of the directive principle and directs the state's responsibility, activities like banning animal slaughter as well as taking care, organizing different animal husbandry measures, okay, adding technology as well as carrying their care on husbandry lines based on scientific means. That is what is there in this directive principle, Article 48. Scientific name of jute is what? Gossypium herbaceum, Triticum avestium, Corcorus olivifolium, Linium usitaceum, and Hibiscus cannabinus. Scientific names, the problem is that you read it how much you want, but you will miss one or other. But the thing is that you should always go through different scientific names. The answer here is C, guys. Okay, it is Corcorus olivifolium. Okay, it is the scientific name of jute. Which of the following are subsidies of NABARD? Very beautiful question. I want to know. I will not answer this. You will answer this. NAB Kisan, NAB Samriddhi, NAB Fins, NAB Foundation, NAB Cons, NAB Ventures, NAB Sanrakshan. You will give answer to this question. I will not give answer. If you don't know this answer, then please, I will request you go through NABARD website very properly. Okay, it is very important that before your phase 1 exam you should go through NABARD website. So the answer is C, which is nothing but 1, 2, 3, 4, 5, 6. All are your subsidies. Okay. Okay, so NAB Kisan, here information has been written, it's a subsidy. NAB Samruddhi, it's a subsidy. NAB Fin, it is also a subsidy. NAB Foundation, subsidy. NAB Cons is also subsidy. NAB Venture is also subsidy. And NAB Sanrakshan is also a wholly owned subsidy of NABARD. So when you are looking at options, here C not, E will be the answer. Okay, C nahi, E rahega answer, that is all are your subsidies. C is not your answer, it's E. ठीक है, किसने किसने C आंसर दिया था? सतविंदर, सहू, विजडम, देन सुष्मिता, गलत हो गया भाई. All are नाब किसान, नाब समृद्धि, नाब फिन, नाब फाउंडेशन, नाब कॉन, नाब संरक्षण, नाब वेंचर्स, सारे आपके सारे के सारे सब्सिडीज है ऑफ नाबार्ड. कैसे गलती कर दी भाई? अंडर RIDF, रूरल इंफ्रास्ट्रक्चर डेवलपमेंट फंड, गवर्नमेंट ऑफ इंडिया क्रिएटेड RIDF in NABARD in 1995-1996. The initial corpus of RIDF fund was how much rupees? Today, it has got allocation in the year of 2022-2023, recently in as of 40,000 crore rupees. But what was the initial corpus? 1000 crore, 2000 crore, 3000 crore, 5000 crore, 6000 crore. So the answer is very simple, that is 2000 crores. Okay. So NABARD 1995-96 got an initial corpus of 2000 crores. It is a food production system that couples water culture, aquatic animals from water, with hydroponics, that is with cultivating crops in water, where nutrient rich aquaculture water is fed hydroponically to plants. Nutrient rich in the sense waste of fishes is provided to the plants. Which type of modern farming methods we are talking? Aeroponics, aquaculture, hydroponics, aquaponics and hydroponics. Answer here is D, aquaponics. Aqua is definitely the aquatic organisms which are in water, and you are then culturing agriculture in your that water medium, aquaponics. Next, it is the organic system of agriculture where farmer focus on maximum yields, first point, limited pieces of land, while at the same time increasing biodiversity as well as maintaining the sustainability and fertility of soil. The Food and Agricultural Organization of United States estimates that only 11% billion, that is 1.4 billion hectares of the world land surface is used for crop production. Therefore, now when we are talking about this type of agriculture, it provides many benefits as compared to the conventional farming and gardening methods. It is inexpensive, sustainable, and can be produced by people who lack resources to implement commercial, chemical and fossil fuel based agriculture. Which is the agriculture we are talking about? Organic, integrated, permaculture, natural farming, bio-intensive farming. So the answer here is which? It's bio-intensive farming. What is bio-intensive farming? When biological inputs are provided, biological resources are provided, biological factors are provided and given as an input to agricultural activity so as to improve its agricultural production, that is called as bio-intensive agriculture. It focuses on maximum yields in minimum area and increasing the biodiversity and sustainability of the factors associated with agricultural production, increasing biodiverse as well as biological resources and inputs in agriculture. This what makes. Which of the following techniques were used for creating first cloned sheep, Dolly? This is the current affairs of the year 2023. So a question can come on this. Which of the following techniques were used to create the first cloned sheep, Dolly? Germinal cell transfer, somatic cell transfer, nuclei transfer, gene transfer and genome engineering. So the answer here is B, which is somatic cell transfer. What is somatic cell transfer? Simple, I will tell you. So what is done is that there is one adult sheep from where nucleus is taken. Okay, here when this donor cells are done from the nucleus, your cell, nucleus and DNA is taken. From other sheep, a egg cell is taken, egg. Now this egg is unfertilized egg. From this egg, the nucleus is removed from the biotechnology. Okay, it may be your biotechnology tweezers, or it may be by picking the nucleus out of the cell. So now the cell becomes without a nucleus. So this is an egg cell without a nucleus. Nucleus is removed. Then after removing the nucleus, now this unnucleated egg cell here is there. In this N-nucleated egg cell, the nucleus which we took from the donor, which is a other sheep, that nucleus is transplanted here in the enucleated cell. So now the cell or the somatic medium in that cell is from a different sheep. The nucleus is of different sheep. Now this hybrid egg is transferred in surrogate mother. So this is a 3-parent baby. Somatic cell from this cell, this sheep, nucleus, that is DNA material guys from this sheep, and the characters and the birth will take from these sheep. So this is a 3-parent baby. This is your somatic cell transfer. This happened with Dolly, the first clone sheep. So in this way, here also you could see Sahiwal cow is there, Gir cow is there. From Gir cow, somatic cells are taken. From Sahiwal, nucleus is taken. Nucleus is added in the somatic cells of Gir cow. That Gir cow's new hybrid egg cell is transferred in other Gir cow, and then first your Dolly was produced. Okay. So first, India's first Gir cloned female calf was born on 16th March 2023. This cow is named as Ganga. This is India's first cloned cow. Okay. Dolly, first cloned sheep in 1996. India's first cloned cow is Ganga. 2023, last year only, no question is asked. Maybe this year the question can be asked. So this is the news, India's first cloned Desi Ganga produced, okay, which is the organization National Dairy Research Institution, Karnal. It produced first cloned female calf, hybrid, which are the hybrids Sahiwal and Gir cow, okay. Next. Yes, so other thing when I am talking about, here also you could see various methods through which transplantation is done. So other options are also there. Let's try to understand. So other thing is gene transfer, in which your genetic material is taken and transferred. Nuclei transfer is nothing but, as I told you, nucleus is transferred from one cell to other. Then is germinal cell transfer, and then is genome engineering. Okay. Then here you could see somatic cells are removed. Okay. And this is nothing but your genome transfer is done. ICAR, Indian Institute of Wheat and Barley Research, is located at which of the following location? So the answer here is A, Haryana. You should know all the important ICARs. For example, this ICAR was related to wheat and barley. It's in Haryana. Your table also has been given. You can go through this table for your more understanding. See, there are huge list of ICARs are there, but you should know at least 10 to 15 important ICARs. Cultivated command area for medium irrigated project is how much? Less than 2,000, 2,000 to 10,000, more than 10,000, 5,000 to 10,000, and 100 to 10 to 100 hectares. The answer here is 10. If you see the cultivable command area for more than 10,000 hectares is for major projects. Less than 10,000 but more than 2,000, that is 2,000 to 10,000 is for medium projects, and less than 2,000 is for minor projects. The upper branchy part of the tree above the bole is called as what? Crown, tree top, canopy, jewel, and coop. The upper branchy top of the tree above the bole is called as what? So the answer is very easy, guys. It is your crown. So this part is your crown. Okay. Overall, this part. See, here crown has been written. So overall, where your complete branches are there and expansion of the tree is there. So this complete picture gives you, this is your bole. Above the bole is the top part of the tree, that is called as crown. Which instrument is used to measure soil moisture? Very, very important. Which instruments is used to measure soil moisture? Pyranometer, pycnometer, tensiometer, hygrometer, and piezometer. Which instrument is used to measure soil moisture? Pyranometer, pycnometer, tensiometer, hygrometer, piezometer. So it is D, that is your hygrometer. If you see pyranometer, it is nothing but used to measure solar irradiance. Pycnometer is used to measure density. Hygrometer is to measure humidity or moisture. Piezometer is to measure pressure. Okay. Cereals like rice, wheat, maize, sugarcane, sorghum are the major staple foods of most of the population. Which of the above given crop has highest acreage in India? So area wise, which is the most cropped crop in the country? Rice, wheat, maize, sorghum, sugar. Very easy, it is rice. After rice it is wheat. So, and after wheat it is maize. Very important, rice is the crop which is cropped largest area in India. Last question. What event served as catalyst to launch Grow More Food campaign in India? Locust attack of 2019-20, Great Famine of 1876, Bengal Famine of 1943, drought in Maharashtra and Bengal Famine of 1770. Here also the answer is very easy. It is C, that is nothing but Bengal Famine of 1943. This is nothing but when in India scarcity of food was there. The Bengal Famine, here the food was not available, and this famine in Bengal took 3 million lives where food was not available, even diseases were there. This forced Indian government to start a campaign, Grow More Food. The entire campaign was directed to improve the crop production, increase the overall tons of crop production. So with this we will end the session. Thank you very much. And one more thing guys, as again I'll end by telling you, if you want such questions but in huge bulk, then the only solution is merit section MCQ batch for NABARD Grade A 2024 phase 1 exam. This is where in one lecture only you will get 100, 200 questions, and we'll finish off 2000 MCQs before your phase 1 exam. It's starting on 22nd of August. Don't miss the dates. Thank you very much. See you next time.